Good morning students. In last video lectures we have learned about the alimentary canal and associated digestive glands. In this video lecture we are going to learn about the physiology of digestion in the alimentary canal. Means the actual process of digestion, how digestion takes place in alimentary canal and which are the different processes taking place in alimentary canal. So we are going to learn in physiology of digestion. So digestion takes place by two ways, mechanical digestion and chemical digestion. Mechanical digestion takes place with the help of mastication, churning, mastication or chewing. Mastication means the conversion of food particles into food into a small particles, that is a mastication, or chewing, which is takes place with the help of teeth. So let's start with digestion in buccal cavity. Digestion in buccal cavity means digestion in mouth cavity or oral cavity. As we know, in the buccal cavity there are teeth, and with the help of teeth mechanical digestion takes place. Teeth converts food particles into a small particles and forms a bolus, and this type of digestion is called mechanical digestion, which takes place with the help of teeth in oral cavity. Now about the chemical digestion. Chemical digestion takes place with the help of enzyme salivary amylase, which is secreted by salivary glands. Salivary gland secretes salivary amylase, which acts on starch, and starch get converted into maltose. The pH for that reaction is 6.8. So in buccal cavity, food particles converted into smaller pieces and forms a bolus, and on that bolus enzyme salivary amylase acts, and carbohydrates, complex carbohydrates or polysaccharide called starch is get digested into a maltose, which is a disaccharide. Now digestion in stomach. As the bolus transfer into the stomach via esophagus, it get digested, or further process of digestion starts in stomach. In stomach there are three types of cells present in gastric mucosa or mucosal cell lining or inner lining of stomach contain gastric glands, and in that gastric glands three types of cells present. First cell is called mucus cell. We secrete mucus. Mucus cell secretes mucus. Mucus is a watery liquid or substance, watery substance. Then there are another cells, peptic cell or chip cell, which secrete enzyme pepsinogen. Pepsinogen is a inactive enzyme or proenzyme. We say proenzyme, which is secreted by peptic cell or chief cell. Then gastric glands also contains a parietal cell or oxyntic cells. Parietal cell or oxyntic cells which secretes SCl. Now what is the role of SCl in stomach? SCl maintains the acidic pH in stomach. It also inhibits the action of salivary amylase. It kills the microorganisms which are entered through food. So these are the different functions of SCl, which is secreted by parietal cell or oxyntic cells. Now the action of enzyme pepsinogen. Pepsinogen is inactive form of enzyme which is secreted by peptic cells. This inactive form of enzyme converted into active enzyme, that is pepsinogen, under the action of SCl converted into pepsin. So there is one more function of SCl, that is it converts inactive enzyme into active enzyme, or proenzyme converted into active enzyme. under the action of HCl. So pepsinogen converted into pepsin at the pH 1.8. Now this pepsin is a protein digesting enzyme or proteolytic enzyme. So this enzyme acts on protein and proteins converted into peptones and proteases. Now in stomach there is another enzyme called renin, which acts on casein. In stomach there is another enzyme called renin which acts on casein. Casein is a milk protein. This milk protein converted into calcium paracaseinate under the action of renin and calcium. This casein enzyme presents in the newborn babies which are feed on milk in early phase of or early weeks of life or early phases of life. Just newborn baby has this enzyme renin, and this renin acts on casein. Casein is a milk protein and it converted into calcium paracaseinate, and this on calcium paracaseinate or coagulation of milk takes place with the help of renin, and this coagulated milk then further digested with the help of enzyme pepsin. So this is the digestion in stomach. In stomach, three types of cells present: mucus cell, peptic cells, parietal cells. Mucus cells secrete mucus, peptic cells secrete inactive enzyme pepsinogen, and parietal cell secretes HCl. So HCl has four roles: killing the microorganisms from food, maintains the pH of stomach, inactive the salivary amylase, and activation of pepsinogen into pepsin. And pepsinogen converted into pepsin, and this pepsin acts on protein, and protein converted into peptones and proteases. So this is about the digestion in stomach. Now after the digestion of stomach, the food passes into a small intestine. So in small intestine, three types of juices takes parts in digestion. First, pancreatic juices, bile juice, and intestinal juice. So bile juice is secreted by liver, and bile juice does not contain any type of enzyme, but it has a role in emulsification of fat. So it emulsifies the fat, and actual enzymes present in pancreatic juices and intestinal juices. So action of pancreatic juices in small intestine. So pancreatic juices contain pancreatic amylase, pancreatic lipase. So pancreatic amylase acts on starch or a glycogen. Starch and glycogens are stored polysaccharides, or they are carbohydrates, polysaccharide. So pancreatic amylase acts on starch and glycogen under the alkaline medium, and this converted into maltose, that is disaccharide. Pancreatic lipase acts on lipid, and lipid converted into fatty acid and glycerol. Pancreatic juices also contain inactive enzyme trypsinogen. The action of enterokinase converted trypsinogen into trypsin, or inactive trypsinogen get converted into active trypsin. Then another enzyme, chymotrypsinogen. Chymotrypsinogen is also an inactive form of enzyme which is present in which is secreted by pancreatic juice or pancreas. Acinar cells of pancreas secretes the pancreatic juice. So chymotrypsinogen is also an active form of enzyme which get converted into chymotrypsin by the action of enzyme trypsin. Now trypsin and chymotrypsin Both are protein digesting enzymes. So trypsin and chymotrypsin acts on protein, and protein get converted into dipeptides. So this is about the digestion in small intestine, where the bile emulsifies the food or emulsifies the fat, and pancreatic juices and intestinal juices acts on food. So pancreatic juices contains pancreatic amylase, pancreatic lipase, enterokinase, chymotrypsinogen, trypsinogen. So these enzymes of pancreatic juices. Then action of intestinal juices. These are also secreted in small intestine. So intestinal juices contain maltose, sucrase, lactase, dipeptidase and lipase. So maltase, sucrase, lactase, these are the carbohydrate digesting enzyme. These are the carbohydrate digesting enzyme. Enzyme maltase acts on maltose. Maltose is a disaccharide, and this maltose get converted into glucose, 2 molecules of glucose. Glucose is a monosaccharide. Enzyme sucrase acts on sucrose. Sucrose also a disaccharide. This sucrose get converted into glucose and fructose, that is a monosaccharide. Then lactose. Lactose is a disaccharide, and on that disaccharide enzyme lactase acts, and lactose get converted into glucose and galactose, and these are also monosaccharides. Then dipeptidase, this is a protein digesting enzyme. This enzyme act on dipeptides, and these dipeptides get converted into amino acids. Lipase is a fat digesting enzyme. This enzyme acts on fat, and it get converted into fatty acid and glycerol. So this is about the digestion in small intestine. Then digestion in large intestine. So some remaining carbohydrates and proteins get digested in large intestine with the help of fermentation by bacteria. So carbohydrates with the help of fermentation by bacteria or bacterial fermentation converted into H2, CO2 and methane, and protein converted into indoles, ketol and H2S. These are the gases liberated by the digestion of protein in large intestine by the action of bacteria which are present in large intestine, and due to that gases the fecal matter get a particular smell by these gases. So this is about the physiology of digestion which is taking place in alimentary canal. Digestion starts in mouth or in buccal cavity. In buccal cavity, enzyme salivary amylase acts, which convert starch into maltose. Then this food or this partially digested form of food transfer into stomach, and in stomach under the action of pepsinogen and casein, under the action of pepsinogen and renin, the food get digested, and this partially digested food enters into the small intestine, where pancreatic juices and intestinal juices acts on the food, and food get digested in small intestine, and the remaining part of the food or some partially digested form enters into the large intestine, and where it converted into carbohydrate converted into H2CO2 methane, and protein converted into indoles, ketol and H2S. So this is about the physiology of digestion in alimentary canal. In next lecture we are going to learn about the process of absorption of digested food. So here I want to also tell you that The process of digestion, or we define digestion, is define digestion in such a way that it is a process of conversion of complex food material into a simple, diffusible or absorbable form. And you can see the reactions which are given in this lecture, that carbohydrates, proteins, fat, these are the complex molecules, and how with the help of enzymes they are converted into simple forms such as a glucose, fatty acid glycerol, fructose, amino acids. So with the help of this chemical reaction or enzymatic reaction, complex food material get converted into simple form. So in next video lecture we are going to learn about the absorption of food. Thank you.